A very good afternoon to everybody. Here is Dr. Priyadarshini, pathology professor from Sri Lakshminarayana College of Medical Sciences. Today, the topic on pathology is blood grouping, which is the most important minor hematology exercise which you all frequently encounter in the practical examination, right? So, we'll be discussing something in relation to theory as well as practical or preliminary. Yes. So, as I've already told, this is part of pathology practical exam, which comes under minor hematology exercise. And I will be discussing little bit in detail about the theoretical part of blood grouping and also regarding OSPE, which is objective structured practical exam. So this is continuation of what you have learned or revision of what you have learned in your first years, right? So the blood group systems are classified into major and minor blood group systems. Uh, The major blood group systems, as you know very well, is of ABO and red system, whereas minor, you have lists of minor blood group systems starting from Kell, Gibb, Duffy, P, LM, Q, everything, right? So, what is the basis behind the blood group system is that you all should be knowing about Landsteiner's law, also called as Landsteiner's principle of blood group system. What does it mean? If an antigen present in the red cell of an individual, then the corresponding antibody must be absent in the serum and vice versa, the condition. To explain this in detail, as you know, the in our body has got antigen as well as antibody. Antigen always present in tissue or the cell membrane, whereas antibodies are produced and secreted in the serum or plasma of individual, right? So when an antigen is in regard to blood group system, antigen obviously is going to present on which cell membrane? Red cell membrane. So the red cell membrane has got antigen, which is blood group antigen, also called as agglutinogen. What happens when an antigen and antibody are present together in a system? Know very well, antigen and antibody reacts or combines to form an immune complex or agglutination of red cells. It makes the cell sticky and allows the adjacent red cell also to stick together to form an agglutination. So when a red cell combines together to form an agglutination, in later stage it is going to get hemolyzed. We don't want that to happen in our system, right? So this is a normal phenomenon or under normal condition in regard to blood group system. When a red cell antigen present in the surface of red cell, then the corresponding antibody, what I mean here is the anti-A antibody, also called as agglutinin, must be absent in the serum. Yes, why? Because we don't want glutination or sticking of RBCs to happen. So, in short, when an antigen present in the red cell membrane, the corresponding antibody must be absent. And the vice versa should occur in the normal condition. So, which is called as Landsteiner law. Who is Landsteiner? You all know Paul Landsteiner, who is the father of transfusion medicine, who first discovered the presence of blood group antigen. So it is also called as Landsteiner law, named after his discovery of blood group antigens. Now that he identified blood group systems, it is divided, I said, into major and minor. I also want to tell about that this Landsteiner law has got an exception in case of RH system. You know what you mean by RH? I'm not taking at the level of first year as you all reached phase 2 and second year. All these are the known terminologies for you. So RH stands for rhesus. Rhesus, as it is first identified in the rhesus monkey. So this RH system has got an antigens named as CDE, right? So here the D antigen is the most clinically significant Rh antigen. Now that we discussed something of Landsteiner law, that when an antigen present, corresponding antibody must be absent in the blood group. But the exception is Rh system, that is when the antigen is present, the corresponding antibody will be absent. Okay? The vice versa will occur in case of Rh system when an antigen is get exposed, that is Rh antigen is exposed, then the corresponding antibody will be present. Okay? When they are D exposed, antigen D antigen is exposed, then the corresponding anti-D antibody will be present. So that is an exception. Now moving on to major and minor blood group. Uh, so I said the major blood group system is ABO and RH. Why it is called major blood group? Because it is seen in majority of our population. So it is called as major blood group system. So as far as you know about your group and your family and friends, whomever you ask, what do they say? What is your blood group? Either A positive or A negative, B positive or B negative, AB positive, AB negative, O positive, O negative. This is what you would have heard from your friends and families. It is very, very, very rare to have other blood groups. So since it is present in majority of the population, it is also called as major blood group system. Are you all following? So ABO and RH is the major blood group system. As I discussed in the prior slide as well, this system was discovered by Carl Landsteiner in 1901. That is why it is also called as Landsteiner's law, because he found out the basis behind the blood group system, that is when antigen present, corresponding antibody absent. So what all antigens he discovered? He discovered A and B antigen. So A and B antigen, if both are absent, he wanted to call it as O. Yes. So A, B, O antigen. The ABO antigens and their corresponding antibodies are present in the following manner in all groups. We take up RBC. You take an RBC. You look at the antigen pattern on the surface of red cell membrane. So if the RBC carries A antigen in the cell membrane, as per Landsteiner's law, what it should have antibody? Corresponding antibody should be absent. So the corresponding antibody of A is anti-A. So it will be absent. But those persons carry anti-B antibody, right? So why? Because if an altered blood group is entered into human system, then it can cause hemolysis. So when a red cell carries A antigen, then obviously the plasma is going to carry anti-B antibody. So the final blood group, what do you want to call it as? A blood group. So remember in short, whatever antigen sitting on cell membrane, that is the blood group. For example, if A antigen is present in cell membrane, it is A blood group, and B antigen is present in the cell membrane, it is B blood group. Can you all follow it up? Yes. So A antigen, A blood group, B antigen, B blood group, and when both the antigens are present on cell membrane, it is called AB blood group. But what you see in between is that the antibody, right? Sorry. So what antibodies are present? You can see A antigen carries anti-B antibody. B antigen carries anti-A antibody. Now that A, B, both antigens are present. So what antibody it should carry? You can see there are no antibodies in the plasma. Whereas the other way, when there is no antigen, you can see there are no antigen here, and whereas both the antibodies are present. Does this fulfill the Landsteiner's law? Yes, of course. So when there is no antigen, you can also remember in such a way there is zero antigen. So it is what blood group? O blood group. Is it all of you are following? So A has been further subdivided into two subgroups, that is A1 and A2, based on reaction with anti-leptin antibody, right? So moving on to the next slide, that is the minor blood group system. What we discussed for major, it is present in majority of population. Same for minor blood group system. Why is it called as minor blood group? Is that since it is present in very minimal population, it is called as minor blood group. To your knowledge, have you heard of anyone who is being called as Kell blood group, Kit blood group? I'm sorry about the first half. Okay, that is not minor. Kell, Kit, Duffy, MNS, Lewis, and Lutheran all are coming into minor blood group systems. Very, very, very rare to find such people. Okay. This RH actually doesn't come under minor, but RHD antigens are present only on red cells and their presence is designated as RH positive and absence is called as RH negative. But I said the exception for Landsteiner's law is RH. So the RH negative individuals become sensitized and with an RH antigen if transfused with RH positive blood or pregnant with RH positive fetus. Okay, so they can get exposed to this antigen. As I told, this is not coming under minor. Okay. I'm sorry about the hiccups happening. So the what are major blood group system? Is ABO and RH. Okay. ABO and RH. Minor blood group is Kell, Kit and Duffy, MNS, Lewis and Lutheran. Just remember that there is something wrong with the slides. So the Bombay blood group is another rarest blood group where there is HH phenotype. You can notice that there are more two small heads present there. This group is characterized by absence of A, B as well as H antigen. You know that H gene is the precursor of all blood group antigens, right? H gene produces H substance, and this H substance combines with various glucose particles and turns into either A antigen or B antigen. When this H substance itself is totally lacking, then what will happen? There is no H antigen and thereby no A or B antigen. So this Bombay blood group is different from O negative blood group because there is also absence of H antigen and presence of anti-H antibody here. Okay, anti-H antibody present over here. So that makes it as different from which blood group you have to differentiate this from? O negative. Otherwise, O negative blood group also has similar pattern except that they contain H antigen and anti-H antibody will be absent. Are you all following? Now that universal blood donor types and recipients. Have you heard of this terminology, universal donors, universal recipients? What do you mean by universal donors? Universal donors are nothing but the donors whose blood is eligible to be given to every other patient. Same way, universal recipients, they are ready to receive any kind of blood if they want in case. As you know, any blood transfusion to be concerned, you have something called group specific bloods which has to be donated or transfused to such patients. For example, A blood group can be transfused with only A positive or A negative based on the RH status. In case if the A blood group is not available, they can go for a second choice of O because O doesn't carry any antigen, but then it contains, remember, it contains A, anti-A antibody. So what makes it to be an universal donor or recipient is that you can note here I specifically mentioned O negative red cells. You can see when the blood is being donated, you are dividing the whole blood into multiple components. Number one is packed red cell. Then you have fresh frozen plasma, platelet concentrate, and cryopreserve. These are four major components. So you have any list of fractionated components available to be produced from the same whole blood group. But then the major division is on a red cell, packed red cell, and platelet concentrate, followed by fresh frozen plasma. Right? So here you can see the O negative red cells. What are the antigens and antibodies present in O negative? O negative has got no antigens. That is why it is called O. As you all remember, lacks all antigens in red cells. That is A, B, as well as D, which is the Rh antigen. So O negative red cells lacks all antigens and are considered as universal donor. Whereas the other way around, AB positive plasma, AB positive blood group individuals are considered as universal recipient. Here it is O negative red cell and AB plasma is considered as universal donor because O negative red cells lacks all antigen, whereas AB negative plasma, AB plasma, okay, FFP, that has got. So this fresh frozen plasma of AB blood group has got no antibodies in them. So both of them are considered as universal donor. Whereas who are all universal recipients given here? You can see AB positive red cells. So what all antigens present in that? A antigen as well as B antigen. So when both are together, obviously A antigens are relatively higher in concentration than B. So in case of non-availability, you can consider these. Okay. So A and B positive blood group individuals can receive blood either from A blood group, B blood group, as well as AB, that is very well known. AB has to receive only AB, but in absence of AB availability, what you can go for either A blood group because it contains A antigen, which is not going to harm. I'm talking about only red cells. Remember that because A blood group plasma should not be given to AB because they contain anti-B antibody. So the other way, AB O blood group also can be given to this AB positive individual. So universal donors and universal recipients, you have to remember, right? So how do you detect blood group and the Rh type? There are two types of grouping systems. One is forward grouping, also called as cell grouping. Other one is reverse grouping, also called as serum grouping. Okay. So now that the forward grouping is the most common technique, which wherever you enter, just they wake up, do a finger prick, take a blood sample and tell you immediately the results, right? So those are considered as forward. What you do in practicals is the forward grouping. It's also called a cell grouping because when you prick the finger and take the blood, it is more rich in red cells. Obviously, it also contains plasma, but more red cells are there. Okay. So what you've been given is the anti-sera bottles. You remember all yellow, white, blue colored bottles, which are all known anti-sera, anti-A, anti-B, anti-D, anti-seras. So known anti-sera with the unknown antigen is considered as forward or cell grouping. Whereas the other way around, when the known red cells, there are known A cells, B cells which are present in the blood banks, to which unknown antibody, if for example a patient's plasma or serum is done, it is called as serum or reverse grouping. Always we should do both forward and reverse grouping together at a time because one step acts as a checkpoint for the other step. For example, in case of mismatch or discrepancy of blood samples, it can be rectified with this. So two blood samples, one for forward one for reverse grouping has to be received, and this forward should match with the reverse grouping. So this is just to reassure or to rule out any discrepancy in the patient's sampling technique or change of blood samples in the ward, right? For example, they send a patient sample. Instead of two, only one sample has been sent. Or even in the same sample, it can be done in the same sample as well, but two technicians must perform individually, one forward, one reverse, so that the results of both of them should concordant with each other to avoid discrepancy in reporting technique. Okay. So just remember, leave along all those practical difficulties for you at this level. At least forward grouping means known anti-sera with unknown antigen. Reverse grouping is known red cells with unknown antibodies or the unknown serum. You can take it in that way. So what is the principle behind blood grouping? Almost the same more or less as that of your Lansdowne law. So blood grouping is done on the basis of agglutination. Agglutination occurs when an antigen is mixed with corresponding antibody. Take for example, when there is known anti-sera, for example, serum is taken and an unknown antigen patient sample. When the corresponding antibody, for example, I add A anti-sera and the patient is of A blood group, what is going to happen? A antigen on red cell membrane is going to combine with anti-A anti-sera and forms an agglutination. So when agglutination is occurring, which means there is corresponding antigen or antibody in the testing sample. So the principle of blood grouping is based on agglutination. Remember that wherever agglutination occurs, it is considered as positive. Fine. So how do you interpret? I make a tile in which you label anti-A, anti-B, and then anti-B, of course. But here I want you to concentrate only on A, B, and O. So I have limited here. So for example, in a column of anti-A, there is no agglutination, but anti-B also no agglutination. Both are negative, both anti-sera, which means O blood group, right? So whereas you can see here, when anti-A is showing agglutination, what anti-A needs to get agglutinated? It needs A antigen. So when the patient is A blood group only, anti-A should be showing some reaction, that is positivity. So you can see here, the blood group is A blood group. Same way the other, anti-B is showing agglutination. So what it is? B blood group. So you can see here agglutination in both anti-A and anti-B, which means it gets agglutinated only when both the antigens are present in the sample. So it is A-B blood. You are following? So now that when all you want to do blood grouping, why I should know blood grouping? Whether you will be doing, you are going to do in this, are you going to be lab technician? Not. But you are going to write the test, right? You are going to write blood group to do blood group test and all. So what is the significance of doing blood grouping? Number one is the most important, which is blood transfusion. There are so many indications of blood transfusion, which we are not going to discuss. But when patient is suggested to transfuse blood, you want to identify compatible blood donors for the patient. So for that purpose, number one. Number two is paternity disputes. For example, to know whether the father is the same or mother is the same. So to rule out paternity disputes, there are pros and cons of that. And obviously, medical legal cases where there are murders, rapes, the blood samples of the victim or the accused, you want to look into that. That also helps. Medical legal cases to prevent hemolytic disease of newborn. We just discussed in RH a few points, right? When the RH negative individual gets exposed to transfusion or gets exposed to an RH positive fetus in utero, they are going to expose to RH and antibody. I said that. But what we discussed, RH is an exemption. So when there is no antigen, ideally as per Lansdowne's law, no antigen, their corresponding antibody will be present. When there is an antigen, the corresponding antibody will be absent. That is Lansky law. But there is an exception for RH. So what does it mean? When an RH antigen or D antigen is present, then the corresponding antibody must be absent. But when they get exposed and gets in contact, what will happen? It is going to cause hemolysis. So when a mother who is Rh negative carrying an Rh positive fetus, there is going to be a condition called hemolytic disease of newborn, because these Rh antigens start traveling from first fetus to the mother, and when the second fetus they get pregnant, these antibodies are going to attack the second fetus. So that is a hemolytic disease of newborn. So that is one of the cause. Then, in knowing susceptibility to disease, there are few conditions, not some are really proven. Like, for example, group A individuals are more prone for gastric carcinoma, and there are some group O blood group, which is more favorite for mosquitoes. So, there's so many studies, all these fascinating ideologies are coming in. So, these are the significance of blood group systems. Sorry, blood grouping. So, what are the methods of detection of blood groups and RH type? Number one is slide and tile type, which all of you should have done in your first years. So, the technique which is being done over a slide or even a big tile. Second is tube technique along the glass tubes. Third is microplate technique, where there is one microplate which is carrying almost 96 wells. So you can do multiple patients at the same go. You can do that, and it is more sensitive as well. And then finally, gel card technique, which is one of the currently done methodology, right? So the gel card technique which we do is one of the most useful and very sensitive method, which you can even store and document the report in the same manner as a gel card. Okay. For your level, at least just know these are the methods. At least four methods you have to know. What are those? Number one, slide, tube, microplate, gel card technique. So, of the list, it is based on the resource setting. I said gel card is one of the currently available technology most of the medical colleges tend to do. But then resource limited settings, the first two are the most common. Number one is slide followed by tube. Compared to slide and tube, which is most advantageous is that tube, I would say. What are the reasons for tube to get higher vote? Is that what are the advantages of tube testing over slide method? Tube, since it is longer and it carries more amount of blood, it allows a longer incubation of antigen antibody mixture. What happens when you mix antigen and antibody? What I mean to say here, antigen in the red cell, antibody in the sera, when you mix in a tiles or a slide, it is easily going to evaporate and dry. It doesn't allow longer incubation for antigen and antibody to react. But in the tube testing, it allows longer incubation time of antigen and antibody mixture without drying the sample. So that is one of the major advantage. Second is the centrifugation. You can put the test tubes inside the centrifuge machine and allows it to mix faster. So this mixing or rotating or centrifugation, whatever you're understanding, this process basically enhances the antigen and antibody reaction. So the antigen and antibody react together to form a centrifugation process, which allows the agglutination faster. If it is a weaker antigen also, it can be identified early. So the weaker antigens and antibodies can be detected because of longer incubation. Same reason. How come weaker is identified in tube rather than tile? Because of longer incubation and centrifugation process, even weak antigens tends to react. And usage of reagents is economized, obviously. And finally, interpretation is more clear because there are no drying artifacts here. So these are some of the advantages of tube testing over the slide method. Yes. So now that we are coming into a practical major thing, we have been introduced for the last 10 minutes. What are blood group system, types of blood group system, Lance-Wiener's law, and what is the Bombay blood group, what are the various methods, what is advantageous method. All these we have discussed, major, minor, RH, antigen, everything. Now that we have come to practical proper. So you should know the procedure. What test are we exactly going to do in practical exam? It is named as forward blood grouping. To understand what you are doing. So it is forward blood grouping by tile method. You know, you all will provided to the tile, right? Label the tile number one. Yes, label the tile with A, B, D. Put a drop of antigen A, which is blue in color, under the A column, and antigen B, which is yellow in color, under the B column, and antigen D, which is colorless, correspondingly in the labeled tiles. So you take a label, sorry, take a tile, label it ABD, and then put corresponding anti-sera as per the, not only for the color, please look into the names written in that anti-sera tube. Then what you have an sera now, you have to add the cells. Now that you add one drop of blood in each labeled zone of the tile. Then always make sure you put anti-sera first and then red cell. Why? Because if you put red color initially, you don't easily appreciate whether you have add anti-sera in that. If you have forgottenly missed in one of the columns, then there will be a wrong interpretation. So now that the third step is mix the cell and region using a clean stick and spread each mixture evenly on the slide over an area of 15 millimeter diameter. So one of the major mistakes done by students is that they are not ready to mix the spread to 15 millimeter diameter. Rather, they make in the same area, they will rotate and the reaction will not be very visible. So try to make the dot or the blood drop into bigger size so that it is easily visible. And another point to be noted is that when you mix the each mixture, you have to separate the stick. You have to use individual stick for one column. Remember that, because what will happen if you use the same stick into the other column, you are carrying an A antigen into the next. So it will be falsely interpreted. So now that rock or rotate the tile. So if not just mixing is not enough, now that you rotate the tile like this, okay, and leave the test for 2 minutes. Here it is given as 2 minutes, but it's not exactly 2 minutes as it is given in our textbooks. But for a minute or so, when you do all these steps properly, then rock again and look for agglutination. Now record the result before trying. Now that this is how you have to interpret. In the tile, in the same way, would have put anti-A, anti-B, anti-D, and there is a control also given. In that control, you will add, instead of anti-sera, here you have blue, yellow, colorless anti-sera. What you add here under control, you will add only normal saline and red cell. So this acts as a negative control. Here there is no agglutination. So you can easily appreciate which is agglutinated, not agglutinated. Now that you have to write the blood group. You will know, you remember the interpretation. For example, I have a positive here, that is the agglutination happens here, and here also agglutination happens. Here there is no agglutination. What do you want to write? It is A, because reaction happened here. So A antigen is present. So A, since D is also agglutinated, you want to call it as A positive blood group. Okay, fine. Yes. So the OSPI checklist, I said this all would be done in the manner of OSPI. Yes. So the OSPI checklist for the forward blood grouping by tile method is, this is the chart which will be kept next to the tile. They will just look into what you are performing. The OSPI is, the examiner is just going to observe what all steps you are doing. Are you doing it in right manner? They are not going to question or viva. This is for 10 marks. So the chart will be kept like this. Number one is perform the forward blood grouping test for the given blood sample with provided items as per the procedure. So the procedure is just 3 marks, whether you are following the checklist or not. That only examiner is going to note down. And after doing the test, now you have to note down the interpretation in the tabular format. What tabular format? We just saw the same way you have to put the tabular format and write the interpretation. What is the given blood group? Yes. So for that, if the blood group is correct and the tabular column is put in proper manner, it is 2 marks. And now that write down the answers for the sub questions given. There will be another chart attached to this, which is carrying 3 questions, and they look for answers for this. So number one is Bombay blood group. Now you know. I'm sorry about Bombay O. It is just a Bombay blood, not Bombay O. So Bombay blood group means what? What all the antigens absent? A, B, H antigen absent. So what all antigens will be present? Obviously, anti-A, anti-B, anti-H antibodies. If you write this book, that is more than enough. Then name the major blood group systems. Very easy. What are those? A, B, O, and RH. Who are universal donors? O, negative red cells. Who are universal recipients? AB positive individuals. Okay. So these are the questions which will be kept for the forward blood grouping. This will be the checklist which will be carried by the examiner. They will just look into how you are performing. Now cleaning and labeling the tile A, B, T and the control. So first you clean, then put label it as A, B, D and control 4 columns for that 0.5 marks. And then adding anti-sera, ABD and saline in 1 drop each appropriately, that is 4.5 marks. And add 1 drop of blood in all 4 compartments, that is another 0.5 marks. Mix the blood and anti-sera with different edges of glass slide. Sometimes they will provide you with applicator stick. So you should each use one end of stick for one mixing and another end for another mixing. That is if they are provided with stick. Sometimes they can keep a glass slides which has got 4 corners. So you can use that also, which is again 0.5 marks. Applicator has to be spreaded over 50 millimeter diameter. Rock the tile relation, that is another 1 mark, because whatever you do here and final agglutination will be more appreciated unless you don't drop the tile. So noting down the interpretation in the tabular format is for another 2 marks. That is the box you have to put and write the interpretation. And writing the correct blood is 1 mark, and the procedure as a whole will be around 5 marks. Now there are sub questions given, which I have already put 2 plus 2 plus 1. So that is total of 10 marks to be given. Now I want to share one video regarding this. I don't know whether it is opening. Yes. So I'm just narrating this. You can see the first step is cleaning the tile. You have to thoroughly clean. Let it dry. Okay. By the time you start adding reagent, it has to be dried because it can cause hemolysis of that. Now we can see it has been put 4 columns, A, B, D, and then control, which she is labeling it as C. Now what you are going to do? Step 2 which you are seeing now, she's going to add, yes, anti-A, anti-sera, which is blue in color. You can notice she's adding 1 drop. Anti-B, you can note it down, which is yellow color, which is she's again adding 1 drop. And anti-D, which is colorless, you can see she's going to add another drop. Then finally, control group is with the help of normal saline. She is carrying in a test tube. We are not showing here the entire saline. So now that the anti-seras are ready, now she's taking the sample. She is trying to mix the sample. That is more important because the red cells would have gone settled in the water. You can see one drop equally added on all. Now that what is she going to do? Now she's taking a glass slide, mixing and spreading over 15 degree. Can you see? She is switching over the edges for each. Can you see? Each edge has been switched over for all four columns, spreaded over, and she's rocking the tile. Can you easily identify which all it is getting clotted? You can see the clot here, you can see the clot here. I'm sorry, it's not a clot, it's an agglutination. Can you see the agglutination here? So what is this blood group here? Obviously A and B. So it is A positive blood group. Is it not easy? So it is reported as A positive. So these are my references. Thank you all for patient listening. Yes. Thank you.